Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 5 potential questions for any Biology Unit 5 paper from Pearson Edexcel. Let's begin with Topic 7. Topic 7, Respiration, Muscles and the Internal Environment. Here I began by looking at respiration in cells. So you need to learn about condensation reactions during formation of ATP, as well as hydrolysis reactions in its breakdown. You need to know that respiration can be aerobic or it can be anaerobic. On this note, you need to learn to draw and label the structure of the mitochondria and include the outer membrane, the inner membrane, the cristae, the matrix, as well as the stocked particles. You need to know the adaptations of the mitochondria for its function, like how it is folded or the infoldings of the cristae to increase the surface area for enzyme-controlled reactions. You need to learn the details of glycolysis until the formation of pyruvate, and you need to know that in glycolysis, 2 net ATP molecules are produced as well as 2 reduced NADs. This is per glucose molecule. You need to know that in absence or in insufficient oxygen, pyruvate is converted to lactic acid in animals and is converted to ethanol in plants. On this note, many questions are asked in relation to how exercise and lactic acid formation affect organisms. The example they can ask about how it can affect their heart rate, the breathing rate. Here, data could be given and you could be asked to describe, comment, or explain. Questions about factors that affect the rate of respiration could also come. Remember, they could ask you about temperature, oxygen concentration, pH, and so on. You need to know carrying out experiments on respiration in a respirometer. And here, remember that potassium hydroxide or soda lime is going to be used to absorb carbon dioxide. Although this is mainly a Unit 6 question, it could come in Unit 5 as well. You need to learn the details of the fate of pyruvate in sufficient oxygen, meaning if aerobic respiration is to take place. You need to know that it goes through the link reactions, the Krebs cycle, and as well as the electron transport chain. You need to know that the link reactions occur in the mitochondrial matrix and learn how acetyl-CoA is produced. You need to learn the details of the Krebs cycle, and you know that it occurs in the matrix. It produces reduced FAD, reduced NAD, carbon dioxide, ATP, and so on. And you need to know that produced reduced NAD and reduced FAD move to the electron transport chain to deliver the electrons as well as the protons in order for chemiosmosis to occur. You need to know the details of chemiosmosis because this is very common. You need to know that electrons move through the electron transport chain and they release energy, and this energy is used to pump the protons from the matrix into the intermembrane space. An electrochemical gradient is going to be created as the protons accumulate in the intermembrane space. A concentration gradient as well as a pH gradient could cause the protons to return, and they will return through ATP synthase enzyme, and this is going to catalyze the formation of ATP. Here, oxygen will be the final electron and proton acceptor, and water will be formed. You need to learn briefly about how respiratory substrates are produced, as seen in Figure A on page 179, and fully understand about calculations for the respiratory quotient. It has been examined before. Moving on to muscles, you need to learn about the different types of skeletal tissues, mainly tendons and ligaments, and you need to know their functions. You need to know that cartilages are shock absorbers in joints, and you need to know about flexors and extensors, although this is not common, but they could ask something about that. You need to know the 3 types of muscles. This is common. You need to know skeletal muscles, cardiac muscles, and smooth muscles, and where they are found in the body. You could be asked to compare and contrast the structure and function of skeletal, smooth, or cardiac muscles. So you need to know how the cells are arranged in these muscles, and you need to know how they adapted for their specific functions. You must know that the muscle contains bundles of fibers, and in each fiber is a muscle cell, which is multinucleated and contains myofibrils. And you need to know that a single myofibril contains a sarcomere. You need to remember that contraction of the muscle in the exam is studied when they observe what occurs in the sarcomere. So please learn the details of muscle contraction, including the role of proteins actin and myosin on page 190. This is a very common question. And then you need to revise about slow twitch and fast twitch muscle fibers. This is also very common. So questions will ask you to compare and contrast slow-twitch muscle fibers, and fast-twitch muscle fibers. And on this note, they usually bring questions about marathon runners as well as sprinters. So you need to know sprinters have more fast-twitch muscle fibers and lower or less slow-twitch muscle fibers, while the marathon runners have more slow-twitch muscle fibers than the fast-twitch muscle fibers. You need to remember that myoglobin has a high affinity for oxygen, so the myoglobin-oxygen dissociation curve is to the left of that of hemoglobin to take up oxygen from it. Please focus a lot on questions on comparing and contrasting slow and fast-twitch muscle fibers in terms of their structure as well as their function, so you need to fully understand. When we go to the cardiac cycle and the heartbeat, we need to know that the sinoatrial node generates a wave or waves of electrical excitation that maintain the intrinsic rhythm of the heart, and these waves spread to the atrioventricular node, but there is a momentary delay that occurs before they spread to the bundle of His, and finally they go to the Purkinje tissues, where this is going to spread along the ventricles. Learn to interpret the electrocardiograms. This is also common. Fully understand the shape of the PQRST waves. Questions asking to comment or explain the changes that occur to the trace during exercise, or if somebody has a specific sickness, could be asked, so please ensure you fully understand that. They could also ask you about how exercise affects the cardiac cycle. Fully understand that. When we move to homeostasis, learn that negative feedback works against the deviation or change from norm, while positive feedback works to intensify the change. You need to know the details of the nervous control of the heart and know that there are receptors around the heart muscle and in the blood vessels, including the aorta and the carotid artery, and these detect the changes that occur either in pressure, pH, heart rate, and so on, and they send impulses to the cardiovascular control center in the brain's medulla, and the response could be carried out either through the sympathetic or the parasympathetic nerve, releasing a neurotransmitter, which could be acetylcholine or noradrenaline. You also need to know the hormonal control of the heart, like adrenaline can bind and stimulate the sinoatrial node to cause increased frequency of excitation and therefore increase the heart rate. You need to know that the volume of blood flowing to various organs can be reduced or increased during exercise. For example, blood to the muscles, the heart, and the skin increases in volume to provide more oxygen and nutrients for aerobic respiration. When we go to control of breathing during exercise, you need to remember about the components of the lung volume, like tidal volume, inspiratory reserve volume, expiratory reserve volume, vital capacity, residual volume, and total lung capacity. Questions can give you data to show the spirometer trace and ask you to calculate specific lung volumes or comment on the changes. You need to remember that a spirometer can be used to produce a spirometer trace, and also remember to describe how a spirometer is used. This is the common question, I've seen it somewhere. You need to know that they use pure oxygen in an airtight chamber, and soda lime, or you could say potassium hydroxide, is used to remove carbon dioxide. The mouthpiece has to be sterilized, and so on. You need to remember how to calculate the ventilation rate, which is tidal volume times the frequency of inspiration. They can ask you about details of how the nervous system controls ventilation. You need to remember that it occurs in the ventilation center in the brain. There is an inspiratory as well as an expiratory center, and these receive impulses from sensory neurons attached to the stretch receptors that could be found in the bronchi. When they send impulses, these causes a response is sent back to the specific muscles of the diaphragm and the intercostal muscles to respond appropriately. Moving on to homeostasis and hormones, you need to remember that hormones are produced from endocrine glands directly into the blood, and the endocrine glands include the pituitary gland, the pancreas gland, adrenal glands, thyroid glands, parathyroid glands, ovaries, testes, and so on. You need to know that hormones participate in homeostatic control, and when a change is detected, the impulses are going to be sent to specific glands and hormones will be produced. Of course, this is going to be through protein synthesis. And the hormones act on target organs to cause a change. So remember the details of negative feedback, mainly in blood glucose control. You need to know that the pituitary gland releases 6 hormones. They are those released from the posterior and those released from the anterior. So we have thyroid stimulating hormone, the growth hormone, adrenocorticotropic hormone, we have the luteinizing hormone, follicle stimulating hormone, as well as prolactin. Those 6 hormones are released from the anterior pituitary, and the 2 hormones, oxytocin and ADH, are released from the posterior pituitary. And on this note, you need to know that actually ADH is not produced in the pituitary gland. It is just stored there. However, it is produced in the hypothalamus. So you fully need to understand the details of how hormones work in causing a change. For example, they could enter cell, binding onto a receptor, and entering the nucleus with the receptor they're binding on, and act as transcription factors. This is common with steroid hormones. For mainly peptide hormones, they bind onto the receptor on the surface of the membrane, and they cause generation of a second messenger, and then other reactions are going to occur. Effect is going to be carried out. Learn to label the kidney, including the renal artery, the renal vein, the cortex, the medulla, pelvis, ureter, bladder, urethra. You also need to know to label the nephron, know the details of ultrafiltration, selective reabsorption, and you need to know the details of the loop of Henle and the importance of the countercurrent multiplier in urine formation. You need to understand how the proximal convoluted tubule is adapted to perform its function. This question is common. You need to know that it's folded and possesses microvilli to increase the surface area, and it has numerous mitochondria to provide energy for active transport. It is surrounded by numerous blood capillaries to increase the concentration gradient. And also remember the details of how ADH takes part in osmoregulation. This is a common question. Remember to include the osmoreceptors in the hypothalamus that detect the change in iron concentration. They send impulses to the pituitary gland that releases the ADH. And you need to remember that ADH is going to travel through the blood and go to the kidneys, where it's going to bind on receptors. When it binds onto those receptors, either on the distal tubule or the collecting ducts, ADH causes the release of a second messenger, which causes the vesicles containing molecules that act as water channels to fuse with the membrane. These molecules will be inserted, and then the membranes will be permeable to water. If an iron concentration is returned to normal, there will be fewer impulses sent and less ADH will be released. Fewer transport molecules will be inserted in the membrane, and less water reabsorption will occur. I recommend that you visit Transport Across Membranes from AS. You need to learn about the structure of the cell membrane, fully understand protein synthesis, and you need to know about gene expression from AS Biology. Moving on to thermoregulation and exercise. Remember the components in the skin that are important in thermoregulation, like sweat glands, the hair erector muscles, temperature receptors, blood capillaries, as well as the shunt. You should know that in warm conditions, the shunt closes and vasodilation occurs. The sweat glands produce more sweat, erector muscles relax, and hairs lie flat to minimize insulation. And in cold conditions, the shunt opens, vasoconstriction occurs, sweat production decreases, erector muscles contract, and the hairs stand upright to trap air and cause insulation. Shivering could be triggered. You need to remember the details of nervous control of body temperature. For example, thermoreceptors in the hypothalamus, as well as those in the skin, detect the change in core body temperature and external temperature respectively. And then the impulses are sent to the thermoregulatory center in the brain, and this will send impulses to organs like the skin, the muscles, and the liver to cause a response that is going to maintain the body temperature. Topic 8, coordination, response, and gene technology. I'll begin by looking at neurons. You need to remember that the nervous system comprises of the central and the peripheral nervous system. The central nervous system is the brain and the spinal cord, and the peripheral nervous system is anything else that is connected to that. You need to know that there are 3 neurons. They are sensory neurons, which are from the receptor to the central nervous system. We have relay neurons between the sensory and the motor neuron, and we have the motor neuron that connects from the central nervous system to the effector, which could be a muscle or gland. This is in case of a reflex action. You need to know that components of a neuron include the cell body, the dendron, the axon, dendrite, and so on. And you need to know that some neurons are myelinated while others do not have myelin. Remember to draw and label the sensory neuron as well as the motor neuron. And remember how impulse is transmitted across them, in which direction it's transmitted. Remember that myelination increases the speed of nerve impulse because conduction will occur at the nodes of Ranvier only. You also need to know that giant axons carry out faster transmission of impulses, and this is a common question. The other common question is myelinated neurons versus non-myelinated neurons in terms of how they perform their functions. You need to know that neurons that are myelinated transfer impulse at a faster rate than non-myelinated neurons. Experimental data can be presented using animals, and questions can be asked in connection with presence of myelin and the size of the axon on the speed of nerve impulse transmission. They could ask you how neurons work. You need to remember the details. For example, how the resting potential is maintained, and this question is very common. You need to know how an action potential is propagated across the axon. You need to know the details of saltatory conduction at the nodes of Ranvier. You also need to remember about the all or nothing law. You need to know about impulse transfer in the axon as well as across the synapse. So for impulse transfer across the synapse, arrival of an impulse in the presynaptic neuron makes its membranes permeable to calcium ions. Calcium ions move into the presynaptic membrane from the synaptic cleft, and they fuse with the vesicles. These vesicles will fuse with the presynaptic membranes. They will release the neurotransmitter into the synaptic cleft. The diffusion of this neurotransmitter will occur across until it reaches the postsynaptic neuron, and here sodium ion channels are going to open, and then propagation of an impulse in the postsynaptic neuron is going to occur. You need to remember that the neurotransmitter is broken down by enzymes once the impulse transfer across the synapse is over. They could ask you about effects of drugs like lidocaine, nicotine, and other toxins on the nervous system. You need to know that some chemicals interfere with impulse transmission by binding onto the receptors of the postsynaptic neuron. This binding could be reversible or irreversible. The next part, they could ask you about sensory receptors and detection of light. Remember that there are primary receptors, and these are directly excited by the stimulus. We also have secondary receptors that synapse with a sensory neuron. A common question can ask about how the generator potential and action potential are generated. This is very common, and you need to focus on that a lot. Remember that generation of a generator potential does not follow the all or nothing law, and if a stimulus is small, a small generator potential will be generated and vice versa. If the generated generator potential reaches a threshold of the specific sensory neuron, then an action potential is generated in that neuron. So for this case, the all or nothing law is going to be followed in generating this action potential. I advise that you revise to label the structure of the eye on page 252 and the functions of each component within the structure. Remember to compare and contrast the structures and functions of rods and cones. This is common, and you need to fully understand the role of rhodopsin. This question is common, and how rhodopsin works. We know that rhodopsin is made up of cis-retinol, and in the presence of light, this cis-retinol is going to be converted into trans-retinol. Then, the bond will break where opsin and retinol are going to be separated. A cascade of reactions take place, and hyperpolarization occurs, generating the generator potential. You need to remember the changes that lead to this hyperpolarization in terms of sodium ion movement across the membrane. When we go to habituation, you need to fully understand what habituation means. Remember the Gill's reflex of the cis lag. This is a common question where data is brought to you from an experiment, and they'll ask you to describe what happens in the Gill reflex. You should remember that calcium ion channels in the presynaptic neuron can become less sensitive due to the repeated stimulation, and this can lead to fewer calcium ion channels being opened, and there'll be less calcium ions moving back into the presynaptic neuron, and this will lead to fewer vesicles being fused with, and there'll be less neurotransmitter released in the synaptic cleft. The excitation potential created in the postsynaptic neuron is going to be lower, and it could be below the threshold, so no action potential will be generated in the postsynaptic neuron. And since this follows the all or nothing law, there will be no response. They could ask about the central nervous system. Remember to label major areas of the brain and their functions, like the pituitary gland, the hypothalamus, the cerebrum, the cerebellum, the medulla oblongata, and so on. Remember about the position of the white and gray matter in the brain as well as the spinal cord. Remember the details of the spinal reflex. This could be common. On that note, remember, a stimulus arrives and the receptor is going to convert that into an electrical signal that goes into the sensory neuron, and then that is going to be delivered to the relay neuron, and the response will be delivered to the motor neuron, and the motor neuron will deliver that to the effector. That could be a muscle or gland, and a response will be carried out. You need to learn to draw and label the sensory as well as the motor neurons. I already talked about this. You need to remember in detail about the pupil reflex. This is also common. Details of how the circular muscles and the radial muscles change on pupil dilation and pupil constriction. You need to remember to compare and contrast the structures of the sympathetic and the parasympathetic nervous systems. On that note, remember to compare and contrast the differences in function between the sympathetic and the parasympathetic nervous systems. They can ask you to compare and contrast the nervous response as well as the hormonal response. You need to know that hormonal response is slower and long-lived, yet the nervous response is faster, short-lived, and most of the time it's targeted. It targets specific organs or specific cells, and those will be the only ones stimulated, while hormones could stimulate multiple areas of your body. Moving on to brain imaging and neurodegeneration, you need to remember the detailed description of methods of brain imaging like CT scans, MRI, functional MRI, and positron emission tomography. Questions can be asked to compare and contrast these methods, and this is a very common question. So you should remember the advantages and disadvantages of each method of imaging. They could ask about Parkinson's and dopamine. So remember details about Parkinson's disease and how it is caused. Remember it's caused by the death of dopamine-producing cells in the substantia nigra. Remember the symptoms of Parkinson's, like tremors, muscle stiffness, and slowness in movement. You should remember that L-dopa or other chemicals like dopamine agonists can be used in the treatment of Parkinson's. And remember that L-dopa can be used because it can cross the blood-brain barrier. They could ask a question in connection to depression and serotonin. You should remember about the selective serotonin reuptake inhibitors and antidepressants that are used. And also questions could be asked about the effect of other drugs on the nervous system. So please be prepared for that. When we go to chemical control in plants, you should remember the interconversions that occur between phytochromes like PFR and phytochrome PR. Questions about the effect of photoperiodism on germination or germination and flowering could be asked as well. They could ask about plant growth hormones and the effects on growth. Here you need to visit page 282 of the textbook. There was a question that came in Unit 5, January 2022. It was question 5B about the effect of auxins on tropisms. So you need to fully understand that because some students found it tricky. You need to know the effects of auxins like indolacetic acid on shoot growth. And remember that some synthetic auxins can be applied as weed killers, so they could bring an experiment on that. Revisit the use of growth regulators like gibberellins in seed germination. Remember this causes the breaking of dormancy, it stimulates elongation of internodes, and promotion of fruit growth. And during germination, remember that the embryo will absorb water, and this is going to cause the release of gibberellin. This will diffuse to the oron layer to produce amylase, and the amylase is going to break down starch as an energy source in the endosperm for the embryo to gain energy and make new cells during growth. You should also know that based on evidence, PFR acts as a transcription factor involved in switching on and switching off of genes. So on this note, they could ask about gene expression that is studied in AS, so please revisit that. When we go to producing recombinant DNA, you need to remember that recombinant DNA is formed artificially by combining genetic material from different organisms, and you should remember the details of making recombinant DNA. This could be a common question, so remember there is making complementary DNA out of messenger RNA using reverse transcriptase, and then using restriction endonucleases to cut target DNA at certain restriction sites. Then there could be integration of DNA into a vector. Some use plasmids as vectors, and then DNA ligase is used as genetic glue. Remember that recombinant cells can be identified using replica plating, so you need to know the details. And since vectors are used as modes of delivery, remember that in animals we have non-viral vectors, which include using a gene gun, liposomes, microinjection, and we have viral vectors whereby harmless viruses are used to deliver the genetic material into the intended cells. You should remember the advantages and disadvantages of using these harmless viruses as vectors. Remember that plasmids are mainly used as vectors in bacteria and plants. You should revisit knockout organisms and gene silencing, and this could be linked to control of gene expression, of course, from AS, where we talked about transcription factors, DNA methylation, histone modification, as well as the role of non-coding RNA. They could ask you about the application of genetic modification in the drug manufacturing, for example, manufacture of human insulin or human growth hormones. And on this note, they could link this to protein synthesis from AS, stem cell therapy, as well as clinical trials. Because this content is talking about drug manufacturing, so when a drug is manufactured, they could ask about clinical trials. Data could be given about plant-based drugs, like making plant-based vaccines through genetic modification, and they could ask you to comment or give advantages and disadvantages, or anything else, maybe explain and so on, so be prepared for that. And fully understand the use of transgenic plants. Remember, they could have a better taste, they could have a longer shelf life, they could be less attacked by pathogens, and so on. You need to revisit the details of microarray and how it's carried out. Remember, these are tools used by scientists to show if a DNA sample from an individual contains any mutation. And remember that bioinformatics uses software and computing tools to organize and analyze huge biological data. Mathematical algorithms and statistical tests are used to interpret data produced from these DNA technologies. You should also revisit advantages and disadvantages of making genetically modified organisms. Experimental results could be presented, and you could be asked to explain the trends in some of these results. So this brings us to the end of these potential questions that could come in any Unit 5 paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.